Yeah, uh, good evening. So, in today's class, we look at sample problems for week 12 of Pharmacognosy and Metabolic Engineering course. The first question is select the bacterial species that are capable of utilizing ferulic acid as sole carbon source. And four options are given here. Pseudomonas fluorescens AN103, Pseudomonas aeruginosa AN107, Pseudomonas fluorescens AN107, Pseudomonas aeruginosa AN103. So this is based on a paper published in the early 2000s where a particular species of Pseudomonas was encoding a few enzymes that could utilize this ferulic acid, and that was shown through a number of assays. You can think about this question for a couple of minutes and then we'll try answering the question. Okay, we start with the structure of ferulic acid. The shown here is the structure of ferulic acid. It has an oxymethyl group and a hydroxyl group here. So this is derived from HBA. It will be converted to vanillic acid. The reaction is shown in the next slide. So what here they observe is that when ferulic acid is present in the growth media of Pseudomonas fluorescens A103, that is the specific species and the right answer. And we have three cases here. First is this um, CoA, which also has a sulfahydryl group, then ATP and MgCl2, but there is no cell extract. The next is CoA sulfahydryl group, ATP and MgCl2, and then we have the cell extract. And finally, we have CoASH, ATP, MgCl2, NAD, and then the cell extract. So these are the three cases. And what they observe is in the absence of cell extract, ferulic acid is present as such. But in the presence of a cell extract, we are able to see vanillin and vanillic acid. Now, how is vanillin and vanillic acid related? Here, we have ferulic acid that gets converted to vanillin and then vanillic acid. This is the probable metabolism that we think is happening within the Pseudomonas fluorescens. They are trying to tell us this is how ferulic acid is getting metabolized within the cell. Further, vanillin, vanillic acid, then we have protocatachoic acid, and then we have ring cleavage, after which we get the carbons. So this is the probable mechanism that's present within the cell. So the answer for this question is Pseudomonas fluorescens AN103. The next question is select the correct substrate that can be used by HCHL to convert it directly into vanillin. The four options are given here: comaryl CoA, ferulyl CoA, caffeoyl CoA, sinapoyl CoA. You can think about this question for a couple of minutes and then we'll come back to it. Okay, so this is a related question. So they mention an enzyme called HCHL, and they say that this can convert something to vanillin directly. What is it something? So HCHL, it can be expanded as hydroxycinnamolyl CoA hydratase lyase. So this will cleave cereal CoA to produce vanillin. So here they say it converts the substrate to vanillin. They don't talk about the other product. The other product is this. So like mentioned earlier here, for the reverse phase HPLC chromatogram, where we had different conditions, this enzyme, the precursor to this, phenyl CoA is produced by this enzyme, where Mg2 plus ATP and CoA which are required. Then phenyl CoA can be directly converted through to vanillin through this hydroxycinnamolyl CoA hydratase lipase. The next question is, identify the false statement regarding Shikonin biosynthesis. An aromatic ring is biosynthesized from shikimate pathway. A pentenyl side chain is attached to the aromatic ring. Carbon-carbon bond formation occurs between the aromatic ring and the prenyl side chain, resulting in the last ring of the quinone skeleton. It is a red-colored compound mainly obtained from the roots of L-erythrorhizin. We will take a couple of minutes to think about this question. We look at each of the given options in the following slides. Yeah, it is interesting. So, we are looking at this question. Identify the false statement regarding Shikonin biosynthesis. Option A, an aromatic ring is biosynthesized from shikimate pathway. Option B, a pentenyl side chain is attached to the aromatic ring. Option C, C-C bond formation occurs between the aromatic ring and the prenyl side chain, resulting in the last ring of quinone skeleton. And option D, it is a red-colored compound obtained mainly from the roots of L-erythrorhizin. Okay, so we look at what Shikonin is. Then we look at each of the options. Okay, so this is the structure of Shikonin. Shikonin, it's a red colored compound. We have other questions where we have figures representing how Shikonin looks like in a solution. So the option B, that is, it's a red colored compound obtained mainly from the roots of erythrorhizin. That is right. You can eliminate that. Next, what are the processes chemically? What are the pathways that are involved in making this Shikonin? From the name itself, because it's derived from the shikimate pathway, half of it is given here, Shikonin. There are two pathways that contribute to it. The phenyl propionate pathway, to the precursor to which is shikimate pathway. And we have the mevalonate pathway as well. So from each of this, one is 4-HPA, the other is geranyl pyrophosphate. So this is 4-HPA, this is geranyl pyrophosphate. From both these pathways, we get 3 geranyl 4-HPA, and then upon multiple steps, we get Shikonin. So this option that the aromatic ring is biosynthesized from the shikimate pathway, this is also right. So this is the aromatic ring, and like how other amino acids are created from the shikimate pathway, here also the aromatic ring is generated from the shikimate pathway. Right. So option A can also be eliminated. Now, a pentanyl side chain is attached to the aromatic ring. So a process called as prenylation happens, and that is this is the prenyl group. This is what gets attached, like you see here in Shikonin. So it's not a pentanyl group. If it's a pentanyl group, you need to have 5 carbons, and uh, clearly, uh, and it can't be like branched here. If it's branched here, it's an iso version. It's an isopentenyl. So this is called a prenylation. So option B, that a pentanyl side chain is attached to the aromatic ring, is wrong. And option C, carbon-carbon bond formation occurs between the aromatic ring and the prenyl side chain, resulting in the last ring of the quinone skeleton. That is true. So this is the carbon-carbon bond where we have the prenyl side group and the aromatic ring. So in detail, I have explained here what happens in this pathway. Right. The next question is, select the correct medium that is standardized by Mitsui Petrochemical Limited in order to induce Shikonin accumulation in lithospermum erythrorhizin cell cultures. Option A, Mg5 media. Option B, MS media. Option C, M9 medium. Option D, LS medium. So you can think about this question for a couple of minutes. It's not very tricky. The two options given here are relevant, but note the key question here. In order to induce Shikonin accumulation, so in all these bioprocessing, two steps are required. One is the step where you grow the cells. The other is the step where you ensure the product is being generated. So both these stages might require different media compositions, because in one you want to just increase the biomass, in the other you want to ensure the enzymes are active and you don't want further increase in the growth of the cells. So think about this question logically, and then we'll discuss it in a couple of minutes. Okay, so like I mentioned here, it's a two-stage culture system. First is the cell proliferation phase. So here the cells are initially cultured in the Mg5 medium for about 9 days to promote cell growth. The next is Shikonin induction phase. The cultured cells are here then transferred to the M9 medium for approximately 14 days, which induces the production of Shikonin. Why M9 medium? So M9 medium is um, uh, not a rich medium, but it's a defined medium. You know what you had there. You have phosphate salts to ensure the pH of the medium is maintained. Then you have carbon source and then a nitrogen source. Um, that's it. So whatever cells that have grown earlier can stay there. They wouldn't grow further. And because during their growth phase itself they have these enzymes produced, most of it would be functional now. That's why M9 medium was used here. Now the question is about the medium in which Shikonin was induced and then accumulated. So that is M9, not Mg5. Right. The next question is identify the reaction that is catalyzed by the bacterial enzyme Coruscant pyruvate lyase. And four options are given here. Coruscant acid gets converted to 4-hydroxybenzoic acid. Coruscant acid gets converted to benzoic acid. Coruscant acid gets converted to 2-hydroxybenzoic acid. And coruscant acid gets converted to 3-hydroxybenzoic acid. You can take a couple of minutes to think about this question. We'll look at the right answer next. Okay, the actual reaction is corismate gets converted to 4-hydroxybenzoate and then pyruvate. So, from the name itself, we know that corismate pyruvate lyase. A lyase would split. So here, corismate pyruvate lyase is split to 4-hydroxybenzoate and then pyruvate. So it is option A. So one additional information is that this is important in the biosynthesis of ubiquinol, which is the coenzyme Q in bacteria. Right. The next question is a strain of Pseudomonas fluorescens AN103 is capable of growing in ferulic acid as the sole carbon source. Utilizing ferulic acid, it produces two major flavoring compounds by degradation of the substrate. Identify the pair of compounds. Option A, vanillin and vanillic acid. Option B, cinnamic acid and hydroxycinnamate. Option C, eugenol and isoeugenol. Option D, caffeic acid and vanillin. So, you can take a couple of minutes to think about this question. We had earlier discussed this in the beginning of today's lecture. We'll go through the process again. Right. So in the first question, we saw that how Pseudomonas fluorescens is able to grow on ferulic acid. So this is ferulic acid, and we have these cases where you have, in the first case, only ferulic acid. In the second case, you have both vanillin and vanillic acid. And in the third case, you have very little of vanillin and more of vanillic acid. In cases B and C, they have, the difference between A, B, and C is that A doesn't have cell extract, but B and C have cell extract. And C, in addition to having the cell extract, has NAD, which is a redox factor, cofactor. So NAD can get converted to NADH. So the whole point is vanillin is the product of ferulic acids, and vanillin can get converted further into vanillic acid. That's what we see here. So this organism, Pseudomonas fluorescens AN03, it would convert ferulyl-CoA or ferulic acid into vanillin. Now, when we have NAD in excess, vanillin itself would get oxidized to vanillic acid, and NAD will get reduced. So that is why you see this saturation for vanillic acid here, the large peak and small of vanillin. So the answer to this question is that vanillin and vanillic acid. The next question is, scientists have successfully transformed HCHL gene to produce vanillin in non-vanillin producing plant. Upon time course HPLC analysis of the plant extract, a gradual decrease was observed in ferulyl-CoA concentration. Identify the correct statement from the following list based on the given information. The expressed enzyme is capable of utilizing ferulyl-CoA acid substrate. The expressed enzyme is not capable of utilizing ferulyl-CoA acid substrate but can produce vanillin. The expressed enzyme is capable of utilizing cinnamoyl-CoA acid substrate. The expressed enzyme is capable of utilizing both ferulyl-CoA and cinnamoyl-CoA acid substrate. So think about this question for a couple of minutes. We earlier saw what HCHL would do. So based on that, you should be able to answer this. So first we see what this HCHL gene does. And the second part of the question is, if you give it enough time, there is a gradual decrease for ferulyl CoA. So what does this mean? Okay, so HCHL stands for hydroxycinnamal CoA hydratase lyase. It produces vanillin in non-vanillin producing plant. So it converts ferulyl CoA to vanillin. That is clear. They say there is a decrease in ferulyl CoA concentration. That is because the substrate gets converted to the product. So ferulyl CoA here, it's the substrate. So among these options here, option A, the enzyme expressed enzyme is capable of utilizing ferulyl CoA as a substrate is the right answer. Right. The next question is, chiconin is a red dye naturally produced in flowering twigs and root of Lithospharum erythrorhizin. Identify the most suitable choice for industrial production of this red pigment from the following list. Shoot culture, hairy root culture, adventitious root culture, and cell suspension culture. Think about this question in terms of industrial production. If shoot culture, hairy root, or adventitious root, or cell suspension culture, which of these methods could be easily scaled up? That would clearly tell you an answer. I mean, we can discuss this as well. Okay, so at the industrial scale, this process happens at two steps. So this has been described briefly in this paper, the link to which I have given. So normally this can be produced in the root, but in the industrial scale, it is very difficult to be able to culture or have scale up, because of which suspension culture is preferred. what you see here is um, the plant Lithospermum erythrorhizum. Here, the flower is white in color, but the root is dark red to dark purple because of the accumulation of this chiconin and its derivatives. Now, if we see here in B, there are two different mediums. One is the cell suspension culture in M9 medium, which produces chiconin and its derivatives. And the one on the right is the LS medium, which is used for cells of culture. So this is how the process is. First, cells are grown in MG5 medium for 9 days. Then they are exchanged into the M9 medium, where they do not grow, but they rather are stably maintained with the necessary carbon and the nitrogen sources, because of which they are able to produce this red color pigment called chiconin. So the answer for this question is option D, cell suspension culture. The next question is, find out the most suitable gene delivery method for plastid transformation from the following list: agrobacterium-mediated gene transfer, adenoviral-mediated gene transfer, particle bombardment via ballistic gene gun, ultrasound-mediated non-viral gene transfer. So this question is specific to gene delivery in plastids and uh, not plant cells alone. So think about what plastids are and why particular methods here will not be suitable. Okay, um, we'll we'll start with describing what plastids are and what are the few prerequisites that are needed for this kind of transformation. So plastids are these organelles in plant cells. These can further be subdivided into chloroplast, chromoplast, and amidoplast. Each have their respective functions. Now, to be able to do plastid transformation, what we need to do is we need to put our gene of interest into the specific vector which can stably survive within plastids and also have homologous regions or flanking regions that would allow for homologous recombination. Now, if we look at the methods presented here, first we should be able to exclude a few methods. For example, adenoviral mediated gene transfer. This works only for mammalian cells and not plant cells. Plant cells are eukaryotic, but they are not mammalian. So option B can be eliminated. The next is agrobacterium mediated gene transfer. Now this has been very well utilized in plant cells, but for uh, introducing this into the genome, but not into the plastid's genome, the nuclear genome. These are different things. So plastid also has specific. So plastids can independently express genes and proteins because it's a tissues, and um, this doesn't correspond to the expression of the genes within the plant cell. So it can independently do it. An agrobacterium mediated gene transfer is a method where you predominantly use it for nuclear method. In addition to that, there are a few plastic targeting sequences like HSP70 or the Rubisco small subunit. These also need to be co-expressed. Without these signals, the proteins will not be directed to the plastids and may end up in the cytoplasm. So, uh, cells that are rich in plastids are taken and they are bombarded with the bialistic gene. Here, what happens is your DNA is encoded with, not encoded, it is coated with gold and it is shot at a very high speed in such a way that this can uh, enter the plastids. Right. The next question is, during the production of human somatotropin hormone in plants, another gene was fused with HST to ensure proper amino acid sequence at the end terminal of the final protein. Identify the second protein. So, four options are given here: helicase, invertase, lipid transfer protein, and then ubiquitin. So, this question deals with expression of a heterologous gene, that is, a human gene within plants. So this would involve thinking with respect to protein folding in a different system. So think about what proteins could stabilize protein folding, and that might help reaching the right answer. Okay, so among the proteins given here, the protein that is easiest to express and expresses very well is ubiquitin. We shouldn't be aiming for function, and here it's given as helicase, invertase, lipid transfer protein, and others. What ubiquitin does is it is very soluble. This would improve the solubility of both uh, R protein because it is going to be fused with the target protein, which is human somatotropin. Now, this protein is from humans and it is expressed in a system which is not humans, that is plants. So, because of the nature of the system, this protein might miss out on post-translational modifications. And because it might miss out on these post-translational modifications, it might not be stable, which would lead to aggregation and misfolding, which is why expressing it with a highly soluble protein like GST or in this case ubiquitin would help in proper folding and prevent them from accumulating in inclusion bodies. So that is the goal of using this with ubiquitin. Right. So with this, we come to an end of this week's problem solving session. I will stay on the call. If there are any doubts, you can ask me.